Thank you. Aloha. Happy Earth Day. Thank you for coming today. And my name's Julie, as John said. Thank you for the introduction. I'd like to thank my advisor, Jonathan Price, who's been a huge help throughout this whole process. Feel free to come in, have a seat. And Flint Hughes is on my committee from the Forest Service. I'm really grateful for your guidance and help. And Jan Schipper, who was with Big Island Invasive Species, has now relocated. He's also on my committee. He's not here today.
Jan did call in, so you're speaking of you today.
He did?
Albizia is here to support you.
Oh, thank you. And that's Springer, our new head of BIISC, the Big Island Invasive Species, who is our newest member. And so today I'm going to be presenting my experience with my internship and also some of my Albizia mapping research that I've been doing for about the past 2 years. And as John said, I started in Michigan. It's my home state. So I started my undergrad there in Traverse City. And I did 2 years there. And I did an internship with a watershed-oriented organization called the Conservation Resource Alliance. And then I transferred to Seattle and did environmental science, resource management, streamside studies minor. And I did a 3-year internship, which was paid with the city of Kirkland in public works and engineering and surface water. And so I got to do a lot of things with civil government and my biggest passion, plants, so doing restoration, streamside restoration, planting rain gardens. And then I started the graduate program at University of Washington in restoration ecology and did 1 semester there and came for a visit to the Big Island and fell in love with this area and knew I wanted to finish graduate school here. So that's kind of how I evolved to this program. And now I have an internship with the Big Island Invasive Species here that I'm going to share with you today. And I knew it would be a good fit because the BIISC is what the short term, what we call Big Island Invasive Species. So I'm just going to say BIISC from now on. Their mission includes education, early detection, control, and rapid response to control the invasive species that are threatening our agriculture or our native ecosystems on Hawaii Island. So I'm going to just go over the internship experience and then talk about some of the issues associated with Albizia and show you my distribution map, my change detection maps, and then I'm going to plug in some of that data into a model, a logistic growth population model, and try to project the growth into the future. So I choose to work with BIISC because they currently hold the front line of defense against invasive species on this island. And my goals for mapping Albizia fit really nicely with the mission of BIISC. And so during this internship, which was about a year ago, I developed a method to map the spatial distribution using remote sensing software, which is ENVI, and ArcGIS. And to produce spatial maps, and then also to try to look at temporal change over time. And so mapping invasive species is necessary because we need to collect the baseline data and also to quantify the change over time. And so also we can also use these maps to look at, better understand life cycles and the dispersal patterns across the landscape. So BIISC partners with private citizens, community groups, businesses, landowners, state agencies, and particularly the DLNR, Department of Land and Natural Resources, and Department of Forest and Wildlife. So this internship provided me a really nice opportunity to kind of understand the complexities of that interagency collaboration. And the main objectives were to make these maps for Albizia, but it turned out to be much more enriching than just that for me. So I was able to work with various agencies. I think the highlight for me was actually walking in the Merry Monarch Parade and with all the other watershed alliances and feeling that sort of camaraderie. And I attended an Albizia removal workday with the Piohana community in Hilo and working with James Leary and trying to understand some of the issues that the community has with Albizia in their neighborhoods. And I also was able to attend a Department of Transportation meeting where we were trying to teach the road crew how to identify invasive species and how to prevent from spreading them. And that was a really nice example to show me how actually challenging it is to prevent the spread of invasive species at an island-wide scale. It's a lot harder than it sounds because the seeds are so small and things can just move around so quickly without us really even realizing it, and the tires. And so this gave me a really nice experience just on the ground, like the reality of doing environmental work and how you actually get the work done. And then I also was able to get some basic aviation training through the Forest Service and the Department of Interior, so I can go up in a helicopter. I hope I get to do that. I haven't been able to do that yet, but I'm trained to do that. And we also were allowed CPR training. So it was a great experience. And I think the biggest thing for me is this Alula Bay ancholine pond restoration project and mangrove removal. This slide is showing the site, and we're in the process, thanks to Anne and Malama Opuna and Renee. They've partnered with BIISC, and they've been really the force behind this project. And we're manually removing these mangroves, no chemicals, and we're uncovering these ancholine ponds. It's in Kona, next to the Honoka'au Harbor, where it's like the dog beach. It's known as the dog beach. And so with my water quality background, I was able to get in there and sample. We did 3 pre-samples, and now we're in the middle of the during removal phase. So they've removed all the big mangroves, and now they're all the little keikis that are sprouting out. And so it's going to take a lot of community volunteer effort to keep it under control. But there is motivation in the community to do so. And we've uncovered a heiau, and so it's a pretty neat project to witness. And it fits nicely with my goals of having an environmental consulting business, because it's giving me like a real project, collecting real data, and to build up my resume at the same time. So I've been hired by BIISC to finish out this grant. It'll last about 7 more months of the post-treatment water quality data. And then I'll look at the data, try to see what kind of effects those mangroves have had on the water quality and also the biologic activity in those ancholine ponds. Another cool thing is we can use the UH Analytical Lab right here in the Marine Science to run the water samples. So we have a YSI probe that we can take in the field measurements, and then I'm measuring salinity, dissolved oxygen, temperature, conductivity, pH, and total dissolved solids. And then what you see me doing there on the right is we're filtering the water samples, and then we send those filtered samples to the lab, and they can run nitrite, nitrate, phosphate, silica, and dissolved organic carbon. And so it's a pretty nice relationship to be able to use our local lab, not have to send it far away, get results quickly. And I used to work in that lab too, so I have a lot of respect for that lab. So now I'm going to switch gears, and we're going to talk about Albizia and some of the background of Albizia and the effects that it's having on the landscape. So the photo on the left is from the early 80s. You can see the one Albizia circled in red. It's along the Wailuku River. And the photo on the right is 32 years later. And so it's clearly showing the change of the landscape. It's blocked the view of the mountain, doing all sorts of things that I'll explain in a few minutes. But the relative fast rate at which change is occurring at the landscape scale is alarming, and I feel is the urgency and timeliness of this research. Because our lowland wet forests in Puna, where I live, are quickly being replaced by this fast-growing tree species. So that's what led me to my research question, just how fast is this canopy spreading across the landscape? And as I said before, it's necessary to collect this kind of data so we can try to quantify change over time. And according to the 2005 Millennium Ecosystem Assessment, invasive species are causing the greatest threat to global biodiversity. And they're key factors in global change dynamics. So Albizia is native to the Moluccas, New Guinea, New Britain, and Solomon Islands. And it was brought here in 1917, and it was aerial seeded from airplanes with a mixture of a whole bunch of other species to restore the degraded watersheds because they had clear-cut a lot of the native species. And the rivers were running and the waters were red, and they had to do something quickly to keep the soil. So that's how Albizia got spread all over everywhere. And Albizia changes the ecosystem functioning, composition, and structure of our native forests. It increases input, increases the availability and the cycling of nutrients, particularly nitrogen. And our forests here are limited by nitrogen in the young lava flows. So the quantity and of the biomass and the levels of nitrogen and phosphorus available in the soil and the leaf litter, it's increased by 4 to 55 times that of the natural background rates, like from precipitation, for example, or the atmosphere. So it's pretty drastic change compared to our native ohia forests. It also may encourage strawberry guava to colonize. And once this happens, the levels of light change dramatically, the level of light reaching the forest floor. So Albizia only allows about 21% of the light to pass through, but strawberry guava only allows 5%. So the image on the left is showing you in red the invasive species, and in green are the native species. And these photos are an example of looking up at the canopy of a native forest, ohia on the bottom, and then the Albizia invaded forest on the top. The pictures are from Keohana Forest Reserve in Puna, which is actually one of our most diverse and intact left little reserves in Puna. So as the guava grows within the native canopy, it closes at the mid-canopy level, and it forms a dense layer of tree crowns, allowing only 5% of the light to come through. The photo on the left is along the Wailuku River. The photo on the top is a natural color image with Albizia crowns on the right and ohia on the left. So you can see, and this is what I do in my work, is you can identify Albizia pretty quickly compared to ohia, just based on the shape of the canopy. The middle color is a hypospectral image of native plants in blue and green and invasives in pink, and the bottom is showing a sectional view of the differences in height. So native forest is on the left, Albizia invaded on the right. So it changes the whole structure. So that's a little background about Albizia, and now I'm going to show you my distribution maps. This is a work in progress, and I actually just got some more funding to try to make this an even better map. But it's a really good start. And Albizia is shown in red. And I'm using WorldView 2, which is the first commercial satellite and it has a 1.84 meter resolution on the ground. So I can detect something as small as a 12-foot spread of one tree. So the resolution is pretty awesome. And this is all available up in our lab, the Spatial Data and Analysis and Visualization Lab, right here in Kanaka-Ole Hall, for anyone in the program that wants to use it. So this imagery in the software are being used to classify, visualize, and quantify vegetation data. And defining scale in landscape ecology is critical. And technologies like this are a really nice bridge between global landscape and local scales. It gives us a really nice snapshot on how to look at the big picture. Also, these maps showing growth over time will help resource managers better understand the complex dynamics of these ecosystems and forests and how to most effectively manage what we have left. So here's just showing you one of the images on the top is the true color. That's the WorldView 2 satellite image around the Wailuku River Hilo Medical Center. And then the image on the bottom is showing you after I run classification. And this has 17 different training areas where you train the software to recognize the spectral signature. And it tries to, and it can separate spectral signatures. But the problem is some plants have a very similar spectral signature, and the software can't necessarily tease out everything from everything. So now I'm working on different methods like texture, edge, to try to tease out eucalyptus. The area circled in black is the blow up of those images that you're seeing. But right above there, those two big stands, those are eucalyptus. Those are not Albizia. So that's my goal right now, is to try to get the software to not pick up the eucalyptus, but only the Albizia. But right now, I have a pretty good estimate of coverage of around about 39 square kilometers of Albizia within just the Puna and Hilo districts. Just the image you see here is the only imagery that I've currently been working with. But how do I tease out, how do I make it so you're getting a more accurate representation? Well, I have to do a lot of ground truthing. That's really the best thing we can do, is get out there on the ground with a mobile GPS unit, you drive around, you log as many coordinates as you can, bring it back to the lab, upload it, and then look at your classification over the real truth data, and that gives you a lot more confidence. And that's how I've been cross-checking and taking photographs in the field in order to identify what is and what isn't Albizia for that count of 39 square kilometers. So I had to subtract a whole bunch. And you do that with your eyes, your naked eyes too. This is just another blow-up showing you Puna. This is Puna Corners. And I live out there and drive by it every day, and I know this is a really good, accurate representation, and Albizia is shown in red. So it's, and on the map it's circled in black, it's the blow-up. So it's pretty big coverage, and it's growing quickly, and that's what I'm going to focus on now, is trying to figure out how fast it is growing, and maybe how much time we have left before it's just one big Albizia canopy. So I've examined aerial photos spanning from 1965 to 1993, and then I have satellite imagery from 2006 and 2009. So I've combined these, and you can scan the aerial photos, you can bring them into GIS, you can georeference your aerial photo and your satellite image, and you can compare. And so that's what I've done. And with confidence, I could only go back as far as 93. But I think I can improve that too, because some people are kind of coming out of the woodwork now with imagery and fly over old aerials. So we might even be able to get do more accurate change detection with some more imagery. But for now, this is what I have, and I have over the 3-year change, this is comparing satellite imagery to satellite imagery. Albizia gained about 46,000 square meters. So this is a net increase of 46%. And now looking back over a 13-year period from 1993 to 2006, Albizia more than doubles. It's actually 2.5 times the size it was, or a net increase of 141% over 31 years. And then the whole span, 16 years, it's about a 3.5 times increase, or a net growth of 253%. So then I plotted this over time, just in a graph, to show the growth. And as the tree becomes established, the rate of growth and spread increases exponentially. But when you average out the growth rate, it's about 8.5% per year. So now what I thought was, this just happened in the last semester. I took a 1-credit computer modeling class, and it turned out to be a huge amount of work. But I plugged in this data and made a little model with the help of Mark Kimura to try to predict future growth based on that change detection work that I did. So this model can be used to calculate a rate of growth, to better understand maybe the time scale which we have to try to protect our lowland wet forests. And my plans are to keep improving this model, and like even Flint suggested, picking out various areas, making this model more robust so we could apply it island-wide. But for now, this model only applies to this one area along the Hilo Medical Center. And the data I used to calibrate is listed on the left. It's from the change detection study. And I ran the model for 200 years. And then I calculated the carrying capacity. And what I mean by that is the photo on the bottom is I take that image and I calculate how much could Albizia spread in that amount of time. And that's the limit that will allow. So some of the assumptions and sources of error are that the urban area will not increase through 2036. This is a closed system. It only applies to this area. The growth rate is the canopy spreading. It's not growth up. It's how it's spreading across the landscape. And growth was only projected for non-urban areas only. So, and we know it grows in urban areas too, but just to keep it simple. So the carrying capacity was about 1.34 square kilometers. And here's the results from that model. I put side-by-side tables just comparing the model's predicted growth with the field data. And the graph is showing the logistic model overlaid over the linear model using the same data, just to sort of give you an idea of the difference in slope. And a logistic model is much more realistic in this case. The field data is shown in red. In 1993, it was 41,050 square meters. Model predicted 40,925. 2006, the field data was 99,227. The model predicted 110. 2009, 145. And the model predicted 137. So then comparing in percentages, they were pretty close. The field data was 141 over a 13-year time period, with the model, which was 169. So both at, approximately a 2.5 times increase. The model field 46%, the model at 36%, and over 16 years, the models, they're both close to 3.5 times the increase. So I feel pretty confident about this model. And so I went ahead and tried to project in 10-year increments, so from 2009 to 2019, and then 29 and to 39. And it looks like it doubles again in the next 10 years, and it almost doubles again in the next 10 years. And then it starts to slow down, and as it reaches carrying capacity, it's going to slow down because it just can't go anywhere anymore. It's just one big canopy. So the percentages do decrease through time, but naturally because you only have a limited amount of space you can grow in. But out in Puna, we don't, in the real world, it will just keep spreading. So, in conclusion, my hopes are that this model, this data, all of you, we can try to, in building on Jaya's work, Jaya just spent 2 years cataloging inventorying all those forest reserves out there, and we know what the hot spots are for biodiversity, and we can target with these distribution maps hot spots to try to get funding to try to control, instead of wasting money in areas maybe that aren't as important for biodiversity, we could apply it to the key areas like Keohana Reserve or Molamaki, where we know there's still a bunch of rare species in there. They're just about to get swallowed by Albizia. And so that's my motivation and my hope that this research can help that process, because I don't think we have a ton of time, probably in our lifetime, but it's hard to say. So yeah, that's all, and I just want to thank lots of people, and I've had tons of help getting to this point, and that's my research assistant, Kaya. He keeps me happy. And thank you for coming today, and thank you for caring, and happy Earth Day.